Hello students, here I am discussing with you the chemical coordination in plants and how chemical coordination occur. The plant is producing chemicals which are similar to hormones. We don't really call them as hormones, but yeah, they are chemicals that promote growth and they control various activities within the plant. So for example, auxins. Auxins are chemicals which are secreted within the plant and they control cell growth and they trigger cell growth by a mechanism which we need to understand in detail. Whenever growth is occurring, growth of a cell is dependent on two factors. When a tissue is growing, it can grow in size because the individual cells are increasing in number, which means mitosis is occurring. Whereas there is another way of how growth can occur, and that is by the individual cells absorbing more water, and when they absorb more water they grow of a larger size, and when individual cells have increased their size, the overall size of the tissue will have increased as well. So auxins are the chemicals that trigger the growth by the latter and not the former, which means that auxins will promote growth by the reabsorption of water or by sending water inside the cell. The cells keep increasing in size and not in number. So mitosis is unaffected with auxins, but yes, the cell keeps on growing because it's absorbing a lot of water. Now the auxins are of various types. The auxin that's in our syllabus, it's called IAA. It stands for indole 3-acetic acid. Now how indole 3-acetic acid works? Indole 3-acetic acid will cross the cell wall and it will bind to the receptor on the cell membrane. Where is the auxin produced? Where is indole 3-acetic acid produced? This IAA is produced in meristems. Meristems are the growing tips of the plant. They include the tips of the roots and the shoots as well. So they produce these auxins. Auxins, they cross the cell wall and they go and they bind to the receptor. Once the auxin is attached to its receptor, this is the same receptor occupancy. You know the auxin will be of a shape complementary to the receptor here, which is present on the cell membrane. So the receptor and the chemical this hormone here, they attach together, and when they attach together, it triggers some events in the cell membrane. Now there are two proteins that we need to consider here. The first is it will stimulate an H+ pump. This H+ ion allow H+ ions to leave the cell. When H+ ions are leaving the cell, this H+ goes from the cytoplasm of the cell outside the cell membrane and towards the cell wall. In the cell wall, there are cellulose fibers. The cellulose fibers are held by the hydrogen bonds. We know that as H+ goes out, it allows the hydrogen bonds to break. The breaking of these hydrogen bonds allow the cellulose cell wall to become more stretchable or more elastic. Otherwise, we know that the cell wall is not very elastic. So H+ weakens the cell wall. Furthermore, there are proteins called expansins. The expansins are proteins present in the cell wall. They allow the cell wall to expand as well. So both are there. So the H+ ion is doing two functions. Number one, breaking the hydrogen bond between the cellulose microfibrils, and number two, promoting the expansins to get activated so the cell wall can become expanded. Another thing that this receptor occupancy with auxin triggered is this potassium channel. This potassium channel allows potassium ions to enter. So you can see H+ ions going out. What they are doing, in turn, potassium ions start to enter the cell. Now what the potassium ion will do, as the potassium ions are entering into the cell, they reduce the water potential here. It means that the cell starts to become concentrated. This in turn allows water to move inwards into the cell through aquaporin channels. Through aquaporin channels, when water is coming in, water moves inwards into the cells start to become turgid, and as the cell becomes turgid, the cell grows in size. If you recall osmosis, if you recall diffusion, we know that when water is coming into the cell, the cell keeps on expanding unless the cell wall starts to exert the pressure potential, because the cell wall cannot extend further. But in this case, this cell wall is weakened because of the effect of the H+ ions and the activation of expansins. This allows the cellulose cell wall to keep on expanding as the water keeps filling into the cell. The cell keeps enlarging in size, and cell keeps enlarging in size until this auxin stays attached here. The moment this auxin gets removed from this receptor, the H+ ion going out will not be going out anymore. The potassium ion won't be coming in, and water won't be coming in either. So the cell wall will be again getting strengthened because of the hydrogen bonds forming between the microfibrils, and this strengthening of the cell wall will then occur around an enlarged cell. So if we start from the size that was initially there for the plant cell and we compare it to the cell size towards the end, we'll see that substantial increase in the size occurred because auxins promoted cell growth through elongation and not through mitosis. Here we are discussing the effect of gibberellins and how gibberellin cause the growth to occur. Gibberellin, on the other hand, is a hormone that triggers the release, that triggers the growth process to occur by increasing mitosis, which means increasing cell division. Now, there are plants that grow tall. This is the tall pea plant, and there are plants that stay dwarf. So the dwarf variety of plant and the tall variety of plant, the difference is one of them is having active gibberellin, which means it will allow a lot of mitosis to occur, and the other is one is having an inactive gibberellin, or you can say the gibberellin is the gibberellin GA1. This is a particular type of gibberellin which is present here in the tall pea plant and absent here in the dwarf pea plant. This active gibberellin, how is it present here and how is it absent here? So basically, there is a gibberellin precursor molecule present in the plant. The gibberellin precursor molecule is present in the tall plant as well as the dwarf plant, but this precursor molecule needs to be converted into active gibberellin, which is GA1, by an enzyme. This enzyme is functional in the tall plant. The same enzyme is non-functional in the dwarf plant. When this is a non-functional enzyme, the precursor molecule cannot be converted to GA1. When this cannot be converted to GA1, the plant stays dwarf. When this converts to an active gibberellin GA1 form, mitosis keeps on proceeding and the plant starts to grow tall. Now, what is the difference between the functional and the non-functional enzyme? The functional enzyme is coded by a dominant allele, LE. We abbreviate it with a capital letter, L and E. And the recessive allele, which is small le, it codes for a non-functioning enzyme. Now, the recessive allele ends up putting threonine amino acid at the active site, whereas the dominant allele sends alanine at the active site. This is a single amino acid change. You can recall what you already have studied in mutations, that mutations occurs as point mutations, and a mutation can cause codon to change. And when the codon changes, the codon change can allow the wrong amino acid to be placed into the primary structure, in which that's called as the missense mutation. So this is a missense mutation. Alanine was supposed to be there in the active site, but because of a missense mutation, the codon changed, and now threonine is there in the active site. And if the amino acid in the active site changes, the enzyme becomes non-functional. The precursor molecule will be there, but it will not be converted to gibberellin. So which plants can grow tall? The plants can grow tall if they have at least one dominant allele. If they are homozygous dominant for both alleles, or they are heterozygous dominant for the same allele, the recessive allele will be there. But since the dominant allele is there, so the plant will grow tall. Whereas if you just look at this genotype here, this genotype small le shows to you that both recessive alleles are present together. Since the dominant allele is absent altogether, the recessive allele will show its effect, and the protein which will be made will be a non-functioning enzyme because of threonine at the active site. So this is how gibberellin promotes growth by increasing mitosis. And this is the gibberellin that we are discussing, GA1. Another form of gibberellin, it allows seed germination to occur, and that I have discussed in another video of mine. That's all from my side. Thank you so much.